Today, we'll be discussing on the topic of strain improvement of industrially important microorganisms, which is the first topic in your syllabus of semester 1. So, strain means producer organisms, improvement of industrially important microorganisms. So, the background of this you already learned in SYBSC. Industrially important microorganisms can be isolated from several natural sources, then they can be modified to increase their productivity. So, industrially important microorganisms, where they exist, what are the sources of their isolation? In general, these microorganisms are isolated from natural habitats like soil and water. So most of the organisms are isolated from soil and water. There may be other exceptions like oil spill areas, oil contaminated hydrocarbon contaminated soils, or anaerobic digesters, etc., etc., compost. So there may be different habitats from where these industrially important microorganisms can be isolated and brought into the laboratory for their further improvement. So the first step in isolation or to search for industrially important microorganisms is primary screening. You must have gone through the approach or the practical of crowded plate technique. So crowded plate technique is one of the method of primary screening for the isolation of antibiotic producing microorganisms. So primary screening uh, you have learned in SYBSC, I think. Then after the selection of desirable microorganisms through primary screening, then we go for secondary screening. So primary screening we just look for the isolation of desirable microorganisms. What are these desirable? If you are looking for antibiotic producer, if you are looking for vitamin producer, if you are looking for organic acid producer, if you are looking for enzyme producer, so using uh, respective media or methodologies, you can go for the isolation of such organisms. Maybe you come up with 500 or 1000 isolates of the desirable as a desirable producer organisms, but then all these desirable or the producer organisms are not equally they are not having equal industrial potential. So in order to check, the second step is to check their potential for the production of desirable metabolites. So potential is checked in relation to quantitative approach. So primary screening basically is a qualitative approach where you don't measure the amount of metabolite which is produced by the desirable strain. But in the second step, in secondary screening, out of 500 or 1000 isolates, you screen these or you categorize these isolates on the basis of the amount of product produced by the desirable microorganisms. So that is a major difference between primary screening and secondary screening. That is how you produce. And after secondary screening, then uh, so approximately 500 isolates you are having during primary screening. So the number of isolates will cut down to approximately 10 to 20 only. So these 10 to 20 isolates will be then further screened in subsequent processes in the laboratory. So all these isolates will be called as natural isolates, or we call them as wild type microorganisms. So wild type means the natural isolates. Okay, so it will be wild type strains or natural strains. So basically these wild type strains during secondary screening, why we selected these isolates? Based on the yield or based on the productivity. So all these isolates will have higher productivity or higher yield. But again, the question is that these natural isolates produce may not produce the desirable metabolites, desirable products, fermentation products in the commercial, in the commercial, at commercial level. Commercial level means at industrial level. So of course, during secondary screening, we conclude that the desirable organism is producing a good amount of fermentation product, but still that natural isolate is not ready to be used in for the industrial processes. That means whatever the quantity which is required for the industrial production is not achieved by that particular isolate. So comparatively, comparison in comparison to industrial production, the production by this particular isolate is very low, or it is producing the metabolite in low concentration. So therefore such type of isolates are not useful for the use in industries. Then what is the solution for that? The solution is that the productivity or the yield capacity of this particular strains should be increased. That means attempts should be made in order to increase the productivity characteristic of these organisms, which is possible. So the attempts are to be made in order to increase the productivity. So along with productivity, the efficiency is also important. So productivity and efficiency of these wild type organisms or natural isolates, it can be improved by the process of optimization of cultural conditions. For example, in laboratory we can supply a desirable pH, desirable temperature, a desirable set of physical and chemical conditions. That means optimum set of conditions is provided to that particular organism. So optimum means optimum for the production of desirable metabolite. Okay, so this is possible in laboratory studies. Supply of optimized set of physical and chemical environment. So this is how the strain may produce the desirable metabolite in higher concentration. Another approach is to increase the potential of these desirable organisms for the production of metabolites is genome modification. You see that genome or the DNA of that particular organism is responsible for controlling all the activities, all the metabolic activities of the cell, directly or indirectly. That is what we have learned. That means all these biochemical mechanisms, biochemical pathways that leads to the production of desirable fermentation products or desirable metabolites is controlled by its genome. So if this genome is altered or if the genome is modified, then there is a possibility of production of desirable metabolites in excess. So this word is very important. We want increased productivity. That means we want that the organism should produce this desirable metabolite in excess quantity beyond its capability. So this is possible by genomic modification. That is another approach, genome modification to increase the yield, potential yield of the product. So simply what we are learning here is the modification or manipulation or modification of microbes for the overproduction. As I said, the microbial metabolites should be produced in excess. That is called as overproduction. So manipulation of microbes for production, overproduction of desirable metabolites, we call it as strain improvement. That is the simplest definition of strain improvement. Improvement in productivity, improvement means to increase its potential. In another simple words, the wild type organisms are converted into the mutants. So if I replace wild type with inferior producers, so we have learned in the last slide that what is the problem with natural isolates, that they produce the microbial metabolites in low concentration. Okay, so that is not affordable at industrial level. So that is a limitation of natural isolates, so we call them as inferior producers. So wild type organisms are inferior producers, they are producing microbial metabolites in limited concentration, so they should be converted into a superior producer. That means their productivity should be increased by means of genetic modification or genome modification. So we call them as superior producers. So if the genetic modification is done, natural are converted, they are genetically modified, what will be the product? The product will be called as a mutant. That means there is genetic modification. So in simple words, inferior producers are converted into superior producers. So conversion of inferior to superior producers is called as strain improvement, and that is the major objective of strain improvement program. So I'm using the word program because this is not a single step reaction. So it is an event, or it is a program. It is a set of several steps. So that is why it is called as strain improvement program, SIP, to increase the productivity and yield of producer organisms. So therefore, the process of strain improvement, it involves continuous genetic modification. As I said, it is not a single step, it is a series of steps, it is a campaign. Therefore, it involves the continuous genetic modifications of the culture. So the genetic modification steps are repeated, may be repeated several times in order to increase the productivity. So modifications are applied and the productivity is checked. Modifications are applied and the productivity is checked. So this process is repeated several times. So therefore, a single organism may receive mutagenic treatments several times. So this is followed by the re-evaluation of its cultural requirements. Superior productivity is a major objective of strain improvement program. As I said, to increase the productivity, to increase the potential of desirable microorganisms for the production of desirable metabolites is the major objective of strain improvement program. So superior productivity, it can be achieved when maximum amount of precursors or substrates are diverted towards the formation of desirable metabolites, and if the control systems are modified, we'll come back to this to discuss that in detail, the modification of biochemical pathways. So it is obvious, what is our logic? The logic is that uh, when there will be when there will be production of desirable metabolite in excess, only when the the precursors or the substrates which are present in the medium or which are present in the cell should be diverted towards their conversion to desirable metabolites. That means the substrates and the precursors should be maximally available for their conversion to desirable metabolites. So the biochemical pathway should be modified such that the precursors and substrates should be diverted towards the production of desirable metabolite, and therefore, and there is a system in the cell which doesn't allow this particular process of divergence. So what is the system is called as control system, feedback inhibition and feedback repression. So these feedback inhibition and repression systems should be altered or modified. If this control system is modified, then this is possible. What is possible? Diversion of precursors and substrates towards formation of desirable metabolites. Therefore, what is important? The important is knowledge of biochemical pathway. So it is very important to have a knowledge of biochemical pathway and what are the feedback inhibition and feedback repression systems existing or which is controlling, which that are controlling the biochemical pathway or the formation of desirable metabolites. So this biochemical pathway, if you know this biochemical pathway and what control systems are there, we can plan the changes in this biochemical pathway, that the if there is existence of a particular control system, so if you remove that particular control system, then if control system is not there, then there will be production or overproduction of microbial metabolites, that is a desirable microbial metabolites. So biochemical pathway and control systems, the knowledge of these two is essential in order to plan the modifications for the overproduction of desirable metabolites. So what is the advantage of this? Plan means what? Whenever we prepare a plan that this modification should be done, if this control system is lifted, then there are chances of overproduction of desirable metabolites. So all these plannings, when we put it on paper, then it becomes a blueprint. Okay, so for the construction of blueprint, the knowledge of biochemical pathway and control system is very important to plan the changes. So these blueprints, one can predict, we can predict the changes, what are the possible changes, what are the possible modifications, or what are the possible techniques which are available for the overproduction of desirable microbial metabolites. Okay, so this is also useful for the selection of techniques of modification. These techniques of modification we are going to learn in this chapter ahead, or to design a technique to increase the productivity. So our understanding that what is the use of this blueprint is prepared by using the elucidating the biochemical pathways and its control systems. So having a knowledge of biochemical pathways is very important here, so that you can plan the changes, you can prepare a blueprint. And what is the use of this blueprint? The use of this blueprint is you can predict the changes, or there are several techniques which are available for modification. So out of these 4 or 5 techniques, which technique you should use in order to get overproduction of microbial metabolite, that you can plan or select. This is also useful for the selection of a technique of modification. Then microbial metabolites, you must have learned that microbial metabolites are fermentation products. We can say fermentation products or microbial metabolites are of two types: primary metabolites and secondary metabolites. So the definitions of these two you should remember. Primary metabolites are those which are produced continuously during active growth phase. That means they are produced during log phase, or we can call it as trophophase. This is very important. So why these metabolites are produced during this phase, growth phase? This means that all these are essential for the growth. That means all these are essential for the growth of microorganism. That means they are involved in active metabolism. So these are the microbial metabolites essential for the organisms for proper growth. Examples are different types of sugars, organic acids, enzymes, amino acids, vitamins, nucleic acids, proteins, etc., etc. All these are the examples of primary metabolites, those which are synthesized in trophophase or active growth phase. Another is secondary metabolite. Secondary metabolites are formed during the end or near the stationary phase of growth. So After the log phase ends, the next phase is the start of beginning of stationary phase of growth. So that phase is called as EDO phase. That means at the end of log phase, in the beginning of stationary phase. You understand what stationary phase is, where the number of microbes remains unchanged. So during this phase, if the microbial metabolites are formed, they are called as secondary metabolites. In simple words, they are formed microbial metabolites formed during stationary phase is called as secondary metabolite. So these products, so secondary metabolism are, so they don't have any significant role in growth and development, but mind well that these metabolites are produced, that means they are required somewhere. They are not required for the growth and multiplication, but they are required for survival of that organism in particular environment. For example, antibiotics. So antibiotics are produced by the organism in order to inhibit the growth of neighboring organisms, and it is very essential for its survival in the existing environment. Then enzyme inhibitors, then the compounds with pharmacological properties are different examples of secondary metabolites. Then the major objective of strain improvement, as I said, is to increase the yield and productivity of metabolites produced by industrial microorganisms. The microbes produce metabolites in limiting concentration as per their need, as we have discussed earlier. They are never produced in excess due to presence of feedback control. This is very important. Why microbes do not produce microbial metabolites in excess? They will produce microbial metabolites according to their requirements. So whatever the amount is required, whatever the concentration is required, whatever the concentration which is utilizable, only that amount will be produced by the natural isolates. And the mechanism behind that is feedback control system. So thus the strains are modified to increase the productivity and efficiency during the strain improvement program. That is the major objective of strain improvement. But at the same time, you need to remember that improvement of strain is to increase the productivity is the major objective, but there may be decrease in the yield or productivity due to the factors other than the productivity, other than this particular factor. So these other factors should also be considered during the strain improvement program. So, however, there are several properties of the organisms that may affect the design and economics of commercial fermentation process. That means there may be decrease in yield due to the factors, due to the other factors other than the productivity factor. So that will see in this. So properties then productivity are also important. That is the point to be considered, because these properties of the organism may lead to decrease in the yield of product. For example, a strain which is producing a very high level of metabolite, particular metabolite, but it is not suitable for a commercial process if its productivity is extremely unstable. That means the organism is genetically unstable. It is getting reverted back to its natural one or wild type. Or if the organism's oxygen demand is very huge, so the organism is having good productivity, but you have to supply a lot of oxygen in order to make that organism produce the desirable metabolite. Then you are consuming a lot of oxygen there. There will be a huge demand of oxygen which is not affordable by the fermentation industry, or that cannot be satisfied in industrial fermenter. So here, what is the property which is affecting the yield is the oxygen demand. Okay,